Hello, in the last video we talked about what DNA and RNA are, where they're located, and we gave roughly a brief idea of what they look like. And so now we're going to take this idea of, basically we knew the structure. So now we're going to take the idea that we know from the structure and talk about why that structure is important and how we actually use that structure to read information. Because again, as a reminder, DNA is the instruction manual that makes everyone a unique individual, that makes every living organism a living organism. So what is it that's being read? What is it that's being used? It determines what proteins will be made. Like, how does your body know what to make? And so that's what we're going to talk about in this video. It is a little bit of a detailed process. I do, in your notes, you'll notice I kind of break it down a little bit more simply, but in this video, it is a lot more simple than your notes. So hopefully you'll see what I'm saying. I'm going to be following along with the notes as I talk about the pictures that are on here. So that way, hopefully you'll be able to identify the information. But if you don't, it's okay because we will talk about it in class. Please be sure to ask questions. It's so important to ask questions. I recognize that you guys don't like to ask questions in front of your classmates, but this information it can be hard to grasp onto, and I don't want you to go into a test or a quiz and not feel prepared. So please let me know, stop me if you have any questions, or ask me on this video. If the question is asked to you, why is DNA important? I would hope that you would come up with the basic answer of, it makes us who we are. But when we break that question down even further, what we're really talking about is the process of making proteins. Every single decision, every trait about you is all in part due to a protein formation. We have thousands of proteins in our body, and all of these proteins work together to do a certain job, telling your cells what jobs to do. And so I want you to understand that yes, DNA is important, but it's the instruction manual, but if we can't read that instruction manual, there's no way we're making proteins. And if we're not making proteins, we're not doing anything for our body. So I really, really want you to understand that the idea of protein synthesis, or the process of making proteins, is why DNA is so important. You have to read that instructions. You have to make those proteins in order for our body to function. So if you recall from the last video, we talked about what's known as the central dogma of biology, and this is basically the whole core concept of living biology, that what we do on the DNA level influences what we see on the really, really big organism level. And we can break this process down, this central dogma, we can break it down into two cycles. The first being transcription. So if we look at this picture here, when we define transcription, that first part of the central dogma, we are defining this process of reading and transcribing DNA and creating a new sequence known as RNA. So we have to read DNA, and from DNA, we use that language, those A, G, T's, and C's, and we interpret to make what's known as an RNA sequence. And so if we look at this picture, like I said, this is the first part of the central dogma, where we have two-stranded DNA, which we know about. We've made models over those. And then you'll see here, we use this information from this DNA sequence. So you'll see A, T, G, C, so on and so forth. And we create what's known as an mRNA sequence based off one strand of DNA. And I'm going to talk about what that looks like. In your notes, you'll see that I have a lot more information on how this is done. But I want you to understand that we have to read the DNA sequence in order to create that RNA sequence. And remember, DNA is stuck in the nucleus. It's not going anywhere. So we have to figure out how to get the information. So what happens is DNA is unwound, and then we read the information from one side of DNA. So we conserve just one side of DNA. We read that information to create an mRNA sequence. That mRNA sequence is continuously adding letters, so A, G's, T, U's, and then from there, once it's done and cut off, that mRNA sequence leaves the nucleus and goes out into the cell. And that leads us into our second part of the central dogma. But the first part is focused solely on reading DNA, creating an mRNA sequence. There are other details that come into that. For example, how DNA is unwound, what we add to the end of the mRNA sequence, all of those things. But really, I just want you to focus on the idea that we're reading DNA, we're creating an mRNA sequence that will leave the nucleus. However, understand that that mRNA is not picture perfect. It does need to go through some editing process before it actually leaves the nucleus. Before we continue with our central dogma discussion, I just want to talk about the genetic code really quick. In your notes, you'll notice that I talked about the genetic code after the central dogma. It's okay, it's not going to interrupt, but I just want to make sure you remember that DNA and RNA are both nucleic acids. They're both made up of nucleotides. Those nucleotides have three different things. One of those three things is known as a nitrogen base. Those nitrogen bases are all associated with the letter A, G, T, C. And when we're talking about RNA, we're talking about a U. So don't forget those things, because the combination of those letters is what determines what RNA sequencing we make. So letters are combined in groups of threes to form code words called codons. We have talked about codons before, but some of you may have not been in class when we did that. We added those notes at the end. So here we'll see codons again. But each codon codes for an amino acid. And again, that's important when we talk about translation in this next slide. But a reminder, the genetic code consists of nitrogen bases, that A, G, T, C, and U. This is the language of DNA and RNA. When these sequences of nitrogen bases are made up, it determines what protein is made. So if those codons are not read correctly, we are not creating the correct protein. This could be a very, very large problem for your body. So please keep that in mind. The reading of this genetic code is one of the most important steps when it comes to ensuring that the organism is healthy and strong. The second part of protein synthesis, or the central dogma, is known as translation. So again, a reminder, transcription occurs in the nucleus, and this is where DNA is read, and we make an mRNA sequence. The second process is known as translation. This occurs in what's known as the ribosomes. So the ribosomes are also found in cells, but it's no longer part of the nucleus. And this is now where we take that mRNA sequence. So remember, you read the DNA, you got the instructions for the protein that you want to make. That mRNA leaves the nucleus, it goes into the ribosomes. From there, that mRNA is read. Again, it's translated from one language to another. That language goes from nucleotides to amino acids. So basically, from that mRNA sequence, we have the manual to make the correct protein. So there are a couple of different types of RNA. One is known as rRNA, the other is known as tRNA. I'm not really particular about you knowing those specifics, but I do want you to understand that RNA is read, it's translated, and then eventually we go into make and build a protein. So here's that part, this second part of the central dogma known as translation, where I'm taking this mRNA sequencing, I'm reading codon by codon, so triplets of threes, so AUG, CCU, if you're looking at that mRNA sequence. And then from there, I am deciding what codon matches with an anticodon. And basically what this means is that I'm taking the perfect matching pair for the codon, and I'm attaching it to what's known as an anticodon. It's like two different Legos, and they attach to one another. When you look at a Lego, it has like a bumpy side, and it has like a side that you put the bumps in. That's what codons and anticodons are. They're perfect connecting pieces. And when you put those perfect connecting pieces together, we can read for what's known as amino acid. When amino acids combine, they create a protein. So one amino acid is not a protein. 13 amino acids together is a protein. So keep that in mind. As a reminder, transcription. We take DNA, we read it, we make an mRNA sequence. That mRNA leaves and goes to the ribosomes. That mRNA is read, translated into amino acids due to the combination of codons and anticodons. When we create those amino acids stick to each other and continue to stick to make a protein. Really, that's pretty well the gist of the entire protein synthesis process. I recognize that I summarized it in a lot less extensive way, but you do have the general idea of what's going on here. Ultimately, it comes down to creating proteins. Why creating proteins is important is because that's what makes us us. Proteins are the workers of our body. So yes, DNA has all the instructions, but if there are no workers to take those instructions and make them into life, there is no life. So a reminder, DNA is like the CEO. Your proteins are your workers. They do everything. So we have to have the CEO tell the workers what to do, tell the workers what jobs they have. If you have any questions, please make sure to ask me on Edpuzzle. We are going to talk about this quite a bit in the next few days. So please, 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 if you don't have questions right away, ask me when they come to you. I just want to make sure that everybody is on the same page because we will have a test over this. And oftentimes you'll see when you struggle on this, you're not going to be able to get it right away. So you have to keep practicing at this information. Again, ask me questions on Edpuzzle.